Now I'm going to get into something in the deep end of the pond here very quickly. The rib clippings that we took on this tiny little few-ounce, 6-inch 6-year-old, which was a very delicate surgical procedure so that we didn't completely destroy the specimen. It was, we were sweating bullets, I have to tell you, medically. I've done plenty of medical procedures and spinal taps and cracked open chest and chest tubes and everything else, but this is, let me tell you, this thing is so tiny and so fragile but the genetic material was extremely high quality. And the man who did the work on that is Dr. Gary Nolan. Now, he's a PhD professor. He is the Harris Chair of biology at Stanford University. He did his postdoc at MIT. He has invented many of the state-of-the-art genetic processing technologies that have been developed in, Silicon Valley and at Stanford. And he did his graduate work and his PhD also at Stanford. But he is a full professor now at Stanford and has a very large lab. So Dr. Nolan has done the genetic work, and the genetic findings are really confusing. So in lay terms, what we know as of right now is that there is, now first before I go into this, so the public gets a picture, Neanderthals and humans have 99.5% identical DNA. A lot of people don't know that. A chimpanzee and a human is around 96, 97% identical. A mouse and a human is around 70+% identical genes. So when they ran the genetic on this, of course they were using very high-tech computerized systems to compare the genes of this humanoid to humans. Now what's interesting, and I can tell you from some emails I have from Dr. Nolan, the first thing he said is the strangest thing he's ever seen, and then the second thing he said is the damnedest thing he'd ever seen, is that it has DNA, it's male, has Y chromosome, it has a B2A marker, which is a marker for that part of Chile that you would expect to find in the mitochondria of females. Now, this is really interesting with what you were saying about the female tradition in Native American culture. And as everyone knows, I don't know if people know this, that this is not a myth, this is a scientific fact, that in the mitochondrial DNA, every person, every woman in the world can be tracked down to an original Eve, an original female. That is true in the mitochondrial DNA. You can go look it up. Don't take my word for it. Okay, so this gets very interesting, but this particular marker would be for that part of the world. So then it came out that there's something like 99%, which doesn't surprise me since Neanderthals are 99% identical to humans, 99% matching with humans. But there are 2 million base pairs of the genome that aren't matching. Now, this is a very high number. And so that comes to around 10% right now that aren't matching. So if you took humans, or is it 99% plus 10? Now, someone said, well, that can be junk DNA. It is not all junk DNA. And the question becomes, what is that? So now here's the other thing, and this is where it gets to be a real kicker. If you drill down into the known dwarfisms, because obviously if this is, whether it's a year old or 6 years old, you can it's clearly not a fetus, and it's only 6 inches large. It would, if you said it was just purely human, it would have to have some kind of genetic mutation that controls for skeletal abnormalities.
Right.
And to this moment, as of the date that I'm doing this interview, they've looked at all those genes, those mutations aren't there.
Wow.
So what is this thing? Now, here's where it gets even more interesting. If you were to take this and just take a step back, the question becomes, is the material that's showing up as human in this creature, did that come from us, or did our genetics come from them?
I was just thinking that.
All right. This gets into the chicken or egg debate that's going to put any mainstream theologian listening to this interview on tilt, to which I apologize in advance. It's also going to put the scientific community on tilt, because you cannot square, you can't square this circle, because here you have human genetic material, or so it appears, at least by a computerized printout. Now remember, to do this manually, when we did the human genome, it took something like $5 billion in a decade. And we're doing something on this creature, just running it through computerized. Now, it's the state-of-the-art systems at Stanford and elsewhere. There are 3 independent labs doing this that are under double-blind studies by one of the most prestigious geneticists in the world, Dr. Nolan, at Stanford. However, if that is true, but then there's this 2 million base pairs and 10% that aren't matching up, and the genes that normally control for skeletal dwarfism or skeletal, what we call dysplasias or abnormalities, those aren't lighting up and aren't being found. Then how do you explain what we call the phenotype? Now I know this is not a medical interview show, but everyone get educated real quickly here. The phenotype is the physical expression of a gene, okay? Like blue eyes, let's say blue eyes, okay? And there are genes that control for the color of your eyes. Everybody knows that. Now, what's interesting is that the genotype isn't matching the phenotype at all. So this is why this is one of the strangest things, and I think it's going to be years of research genetically to get to the bottom of this. And now, because we didn't gather this little body off of a spacecraft that said, hello, we're from this star system, you're never going to be able to say it's ET. But the question is, what is it? And I'm telling you, as a medical doctor who has been consulting with the top team at Stanford University, nobody knows what it is yet. However, in this part of Chile, there is a tradition that they came from the stars, that there were small people that had landed in the area. And we think that this little specimen, this little body, is decades to hundreds of years old. In other words, it predates the modern era of technology. Because some people say, well, maybe it was a human genetic experiment that went wrong. No, it's not, because it predates our ability to do that. We know that from what's called the DNA clock, the amount of cytosine that falls off of the ACTG in your DNA. You can measure and estimate how old the DNA is, and it's old. It's not thousands and thousands of years, like hundreds of thousands, but it's not modern. It's not recent. And you can kind of control for that, although we haven't been able to do the carbon dating yet because we didn't want to use up all of our material until we got the genetics run down, because you can only take so much of this tiny little body without destroying it. So the question becomes, let's say this is a creature, a little being, a little person, a humanoid. It lived decades to a few centuries ago. It may be pre-Columbian. We don't know.
Right.
How was it kept alive? Because we now know it's not an aborted fetus that was grossly deformed, based on the world's expert in skeletal dysplasias who wrote the textbook on the subject, and the opinion of more than one radiologist. We now know that it's got a lot of the same genes we have, and yet there's 2 million base pairs not matching. We know that even today, now I'll tell you, I've unfortunately had to deliver emergently what's called a precipitous delivery, when someone goes into preterm labor and have to deliver some of these really tiny babies, that are, 30, 40 weeks. But this would be on the order of 15 to 25 weeks, in that range at the most. Even today, in your best neonatal intensive care unit, you couldn't keep this creature alive.
No.
How did it live long enough to develop its long bones and its epiphyseal plates to the point that they are, to where this expert is saying it's 6 to 8 years of age and it's only 6 inches tall? It fits in the palm of my hand. So this is extraordinary. Now, we can't say it's ET, but we can't say it's just a normal human finding. It's an unknown humanoid, which still makes this an enormous scientific exploratory adventure. What we have to do is to go back to the Atacama Desert, and here's why. The man at the Institute in Barcelona that is actually physically holding this creature, this little body, went there with another a scientist. And they went to this remote area, and they were told, as they went further and further into remote areas, that there were urns and materials of more of these little bodies that had been found over the years and were kept in containers. Now, they never found them, but if in fact they are there, and Dr. Nolan has said this at Stanford, if you find 2 of these, game over, because you cannot have something that's this obviously different from a human, that's a human genetic defect, and end up with 2 of them that are because there is no syndrome in all of the medical literature that explains this particular morphology or phenotype or skeletal form of this little Atacama humanoid. There's nothing. And it's been exhaustively looked at by the world's expert in that area, Dr. Luckman. So what Dr. Nolan and I have talked about is that if there's a second one of these found, it's kind of game over. But what we have to do is to try to get back down to this area. Now, of course, to do a proper expedition with ground-penetrating radar and sonar and ultrasound and blah and on and on and on is way beyond the funding means that we have. If someone listening wants to fund and underwrite that, I'm all ears. It's way beyond our ability to do. However, it should be done because this could be an enormous discovery. And it's in an area where the native peoples talk about luminous, egg-shaped and disc-shaped craft coming down and going into the foothills of the Andes.